Dr. David Underwood, who's going to speak to us about the host-microbe interactions in gut homeostasis and pathogenesis. He's associate professor in the F. Jaja Foundation, I probably didn't pronounce that right, Inflammatory Bowel and Immunobiology Research Institute at Cedars-Sinai Medical Center in Los Angeles. He's also director of the Medical Center's PhD program in Biomedical Sciences and Translational Medicine, and on the faculty of the David Geffen School of Medicine at UCLA. Welcome, Dr. Underhill.
All right. Thank you for having me here today. So I'm going to talk about something a little different, the microbiome, which hasn't really come up yet, and its role in gut homeostasis and pathogenesis. We've discussed variations of this slide over the last day and a half here, looking at the microflora that's found in the GI tract, okay, and discuss the fact that the numbers of bugs that we find there are astounding, that they vary from place to place in the gut, that there are zillions and zillions of genes to mine for interesting activities in the gut there, and sort of a growing indication in the field that this microflora in the gut is important for a huge amount of physiology in a person. But what I want to point out about most of the discussion of this microflora is that it's really all about bacteria. This is, I think, a feature of the technical history of where we are. The last decade or so, we've been able to do high-throughput 16S sequencing to identify with unprecedented depth the types of bacteria that are found in the gut, and that has led to really an explosion of our understanding of what this community looks like and how it contributes to health and disease. But the technology is in itself a bacteria-specific technology. We'll discuss a little bit how that's beginning to change. My history is that my lab has been studying innate immunity mechanisms for many, many years. We've been very interested in innate immunity against fungi in particular. And so we got kind of interested in whether fungi would actually be a really important part of this community as well. So a few gratuitous slides here on fungi. First of all, I've seen estimates that as much as 25% of the world's biomass is actually fungal. Okay, there's a huge amount of fungus in our environment. If you read National Geographic regularly, you'll know about the ants that get fungi growing in their brains and become zombies to fungi. The luminescent fungi that you can find in South American rainforests, very cool. If you read the New York Times, or maybe I should say the Washington Post, there are fungal pathogens that are killing bats in North America, killing trees in Europe, and frogs all over the world. So they're clearly having a huge impact on our environment. In biomedical sciences, we tend to think of the fungi that cause diseases, and this is kind of where my interest in fungi has been focused in the past. There are a whole variety of different fungi that cause diseases in people. The most common are probably Aspergillus infections and Candida infections. In fact, systemic Candida infections, even today, even with the drugs we have, tend to be about 50% lethal. I mean, we just don't know what to do with these things. And so I think there's an awful lot of interest really in trying to understand what the real natural host defense is against pathogenic fungi. But what I also want to note is that every organism on the planet has evolved in an environment that's very rich in fungi. And so every organism out here has found a way to survive in such an environment and in many cases make use of each other. There are a whole, I mean, this is just a quick set of examples of fungal symbionts that have evolved in our environment. Many organisms live in peace with fungi. I find this one particularly interesting. The cow rumen has 10 to the 11th organisms per gram in its microflora, and it's already understood that about 8% of that is fungus. Okay? So this really got us to thinking, okay, what about the human gut, or in my case, maybe I'm interested in the mouse gut as a model? Are fungi really an important part of that community? So as I said, the 16F microbiome studies out there don't get me anywhere. Okay, I don't know whether that's bacteria specific. We're starting to do more and more sort of metagenomic studies where we just sequence, say, all the DNA in the microflora and then sort it out and figure out who it belongs to. And these are some older studies. There are some newer ones coming out, but they generally agree on this, and they report something on the order of 0.1% is fungal. All right, so we almost quit at this point, right? Because 0.1% is a little hard to get excited about. It's really a significant part of the community or not. But as the lab started to think about this more and more, we came to a couple conclusions that I suppose if you walk away with nothing else from this talk, is this. When you look at these genetics types of studies, you have to think about what you're really reporting, okay? I'm trying to understand the fungal microflora in the gut and how it interacts with the mucosal immune system, and whether that might be important for disease in people ultimately. And you have to think about this a minute. So the average fungus, which is one genome, eukaryotic cell, is 50 times larger than the average bacterium. So if you're just in the business of counting genomes, right, you're basically underreporting the amount of fungus there by 50-fold. Now, the average fungal genome is about twice as big as a bacteria, so maybe I'm exaggerating a little bit, but you get the point there. Also, I want to note that this fungus is encased in a cell wall that's actually harder to extract DNA from than bacteria. And you have to think a little bit about the efficiency with which you're recovering the DNA from the samples you're getting, because ultimately you're just reporting the DNA you've recovered. As this program went on, we found that if you just take a commercial soil DNA extraction kit and add a little zymolase to it to degrade fungal cell walls, you can increase several-fold the amount of fungal DNA you recover. And so again, I think in a lot of ways this was maybe underreporting the amount of fungus that's really there. And these kind of thoughts prompted us to just go look. So we made some primers against fungal genomic ribosomal regions and just amplified from various areas in a mouse colon, and sure enough, we can detect fungus all over the place. All right? Using an antibody against fungal cell walls, sort of a generic antifungal antibody, looking in mouse colon sections, sure enough, we can see that there's fungus all over. If we look a little more closely, what you'll see is that the fungi here are interspersed with the bacteria. It's just a daffy stain, so you can see the bacterial microflora here. And the fungi and the bacteria are all mixed up together, suggesting to us that they actually share similar ecological niches in the gut. Okay, so I'm convinced that there's fungus there. I'm convinced that there's maybe more of it than we've been thinking about. But I guess from the point of view of my lab, I'm only really interested in this if I have any sense that the immune system really interacts with these fungi in a meaningful way. In order to discuss this, I've got to tell you a couple features of the fungal cell wall. The fungal cell wall is a layered structure here. Two major components of the fungal cell wall are the beta-glucan layer here. It's a rigid polymer that forms the hard shell of most fungi, usually beta-1,3 and beta-1,6 glucan. And covering this very often is a layer rich in nanoproteins lots and lots of mannans. Okay, it's estimated in your average yeast like this, that about 40% of the dry weight of the fungal cell wall is beta-glucan, and another 40% of the dry weight is mannan here. Okay, so glucan and mannan. Turns out in the IBD world, one of the things they look for is serum reactivity against an antigen they call ASCA. Okay, and about half the GI docs you ask have no idea what ASCA is. But the other half will tell you this is anti-Saccharomyces cerevisiae antibodies. It turns out that this is one of the first biomarkers capable of identifying the majority of patients with Crohn's disease. Lots of these patients have circulating ASCA antibodies. I mentioned the antigen. Turns out to actually be mannan. Okay, mannan is common to most fungal species, so the nomenclature is a little bit misleading. It's not an anti-Saccharomyces cerevisiae antibody reactivity. It's actually anti-mannan reactivity. More recently, even anti-glucan and anti-chitin antibodies have also been measured in serum from IBD patients, other components of the fungal cell wall. So this suggests that in IBD, there is exposure, novel exposure to fungi, and this is reported as antibodies reactive against fungal cell walls. We looked at whether we could see a similar thing in a mouse model of colitis, and sure enough, in naive mice, we see very little IgM or IgG in serum recognizing mannan. But if you induce colitis in these mice by treating with DSS, you see the appearance of anti-mannan antibodies. Okay, suggesting that indeed the immune system does interact with these fungi when the gut is perturbed. Another way of looking at whether the immune system was going to be able to recognize the fungi that are in the gut was to begin to use Dectin-1 as a probe. So to tell you a little bit about Dectin-1, it plays a big role from here forward in the talk. Dectin-1 is a C-type lectin receptor. Okay, it's a classic innate immune receptor that recognizes fungi. Okay, the C-type lectin domain on the outside actually recognizes the beta-glucan in fungal cell walls. There's a transmembrane domain and a small signaling domain that through Stark and 6 signaling activates phagocytosis and respiratory burst. This is really the primary activity of the receptor. The receptor is expressed on macrophages and dendritic cells and neutrophils, basically all the myeloid phagocytes, and is responsible for triggering phagocytosis and respiratory burst when these cells encounter fungi. Okay. In some cells, it can also activate NFAT and NF-kappa B, helping to drive inflammatory responses. Okay? And just to illustrate this here, we have a macrophage membrane here. This macrophage is expressing a GFP-tagged Dectin-1. So in green, we have Dectin-1. Red is CD45, which is from another study. And we've got some fungal particles here in blue. What you can see is that when this macrophage encounters a fungal particle, the Dectin-1 binds to it and triggers phagocytosis of this particle, and ultimately killing of the fungus. Okay, and it turns out Dectin-1 deficient mice are very susceptible to a wide variety of those pathogenic fungi I showed you. Okay, so it's really important in mice for antifungal host defense, and emerging evidence is showing that in rare people with defects in Dectin-1, they are also highly susceptible to fungal infections. All right, so that's Dectin-1. So we made a soluble form of Dectin-1, okay, that we could fluorescently label. This fluorescently labeled Dectin-1 recognizes fungal cell walls. This is zymosan, which is Saccharomyces cerevisiae, Candida albicans cell walls, Aspergillus cell walls. These are the sorts of things we were interested in. And so we used this fluorescent probe to look at whether it would also recognize the kinds of fungi that we would see in the gut microflora. And sure enough, when we stained mouse colon sections with this fluorescently tagged Dectin-1, we could find fungi all throughout the gut, interspersed, again, with all of the bacteria microflora there. All right, so Dectin-1, at least as an innate immune receptor, seems to be able to recognize the fungi that we see in the gut. We can also use that as a probe in flow cytometry to look at mouse fecal content and see a peak of material here that is recognized specifically by Dectin-1. Okay, you can sort that out, and you can see that the material recognized here has fungal morphology. And this bit of data, I think, is really, really important. If we look at fecal material here by flow cytometry stained with the soluble Dectin-1 and the antifungal antibody that I showed you, what you see is that both of these probes recognize essentially the same material. So it's fungal, all right? And if we treat these mice with fluconazole, okay, a fungal-specific antibiotic, we find that we can deplete this material, indicating that the majority of the fungi we're seeing here is really coming from a self-renewing live population in the gut. All right, so I'm convinced that there's a lot of fungus in the gut. I'm convinced that at least Dectin-1 is an innate immune receptor that can recognize the kinds of fungi that we see in the gut. And so the question arises, is that then anything that matters? Right? So then in a mouse model of colitis, so it's DSS colitis, we looked at whether there was any effect on lacking Dectin-1 in severity of colitis. Okay? Wild-type mice treated with DSS for a few days and then left to recover generally lose a little weight, experience a little intestinal inflammation, and ultimately recover over the course of this experiment, whereas Dectin knockout mice expressed significantly more severe, experienced significantly more severe disease. You can see this with a sort of the gross measure of just weight loss. The mice get sick, they stop eating, they become dehydrated. You can see it with histology here, where you can see that the colonic architecture is disrupted more severely. Histology score here, analysis of these pictures, you can see that the DSS-induced colitis is more severe in Dectin-1 deficient animals. All right. Another way of looking at this is just looking for inflammatory cytokines in the mucosa of these mice, and TNF, IL-23, IL-17, we see more inflammatory cytokines in the tissue of Dectin-1 deficient animals. If we take T cells out from either the lamina propria or the mesenteric lymph node and activate them, we see that in the Dectin knockout mice, we see more IL-17 producing cells and more gamma-producing cells. Sort of re-stimulation ELISA, we see the same thing. Okay, but basically by every measure of inflammation we looked at, the Dectin-1 mice are experiencing more severe inflammation. We had a lot of hope that that inflammation would have a very particular flavor, that it would be a 17 or a gamma or something very, very specific. In fact, we just saw that it was just a mess. Every measure of inflammation is just worse. So then we had to go look and see what kind of fungi were in the gut. Okay, now I really cared. Okay, there's lots of fungus there. If you knock out immunity to fungi, these animals experience more severe colitis. And so we learned how to do ITS sequencing in fungus and went and looked at the fungi that were there. Importantly, we compared the food and the feces. It's very, very different, much to our relief. Did you know that one of the major additives of mouse chow is yeast extract? That was very scary until we decided that it really didn't matter. So looking at the feces, we can see that the majority of the dominant fungal community here is Candida. Okay, not surprising. That's actually known to be a commensal mucosal fungus. We'll talk about it a little bit more in a minute. There's lots of Saccharomyces in there, mostly Saccharomyces cerevisiae. There's lots of Trichosporon species in there, and a whole variety of others. The diversity is really impressive. We were amazed to see this. There are many, many different kinds. I think we were pretty positive in identifying at least 200 different species, pretty confident in at least 50 different genera of fungi in the gut of these mice. And so there's lots of fungus there. You can look up what the makeup seems to be. But now I'm interested in what they're really doing during disease. Okay, so we're going to talk about a couple in specifically as we move forward here. The majority of the Candida, 90+% of the Candida that we found in our mice is actually Candida tropicalis. Okay, Candida is known to be part of the normal microflora. It's found on mucosal surfaces, primarily skin, sinuses, these kinds of things. Many reports have estimated, at least looking at surfaces in people, that at least 60% of humans are colonized with Candida. So it's known to have that kind of relationship with us. But it can cause pathogenic infections, most commonly in immunosuppressed patients, AIDS patients, cancer patients. You'll be aware of this. And it's this pathogenic role that we typically think of it in. But it is this commensal as well. It's a dimorphic fungus that grows in a yeast form and a filamentous form. We're going to compare that moving forward here a little bit to another fungus that we found called Saccharomycosis fibrigera. This is a common environmental fungus, especially good at degrading starches. It's already used industrially for fermentation of starches to produce ethanol, and it's actually one of the major fungi that are used to ferment rice wine. It also grows in a yeast form and a filamentous form here, and was detected in the microflora of our mice. Okay? So just a quick comment, looking specifically at Candida tropicalis and Saccharomycosis fibrigera, looking at different parts of the gut, from the ileum to the distal colon. You can see that C. tropicalis is more prevalent the farther down the GI tract you go, whereas Saccharomycosis fibrigera is actually more common in the higher parts of the GI tract, suggesting that there's a lot of room for trying to understand the diversity of the fungal microflora in the gut that we actually haven't explored yet. Okay, but does the fungal microflora change during inflammation? And the answer is yes. Just look over here for a moment. These are the Dectin knockout mice. They experience very severe inflammation. You treat them with DSS. When you do that, you see that the Candida population expands. You see that the trichosporon there expands. And these are both opportunistic pathogens, and they're actually expanding. The Saccharomyces cerevisiae population contracts and other things as well. This is all confirmed by PCR here with probes specific to each of these different fungi. So we do see changes in the fungal microflora during inflammation. Now, as Yasmin had discussed and as Ramnik had discussed, we don't know if that's causative. There's no reason to believe that that's causative here. It could just be that like the bacterial microflora, it changes when the intestine experiences inflammation, something to be explored. But we did wonder where the fungi were during inflammation, where the fungi were contributing in any way to the severity of the disease in these mice. Okay? If you look at wild-type mice, the fungi are out here in the lumen of the gut. If you treat them with DSS and induce some inflammation, the fungi, for the most part, remain in the lumen of the gut. However, in the Dectin-1 knockout mice, okay, here, in the untreated condition, the fungi are all out in the lumen of the gut, and they're normal. In fact, we looked really hard trying to find differences in the fungal microflora between wild-type and knockout animals. Don't see it. But we see here that when we induce inflammation with DSS, we get invasion of the colonic tissue here with fungi, and we think this is really ultimately what contributes to the increased inflammation and disease in these mice. In the absence of Dectin-1, the innate immune cells down here in the tissue are unable to clear these fungi as quickly. If that's true, we should actually be able to treat these mice with fluconazole. Remember, that depletes the gut microflora and cure them. And turns out that's exactly what happened. Again, looking here at the Dectin-1 knockout mice, they get very severe disease. Give them some DSS, compared to wild-type animals. But if you treat these animals with fluconazole, their disease gets better, okay, indicating that it really is the commensal gut microflora that's contributing to the severity of the disease here. Interestingly, treating wild-type mice with fluconazole didn't help them out any. You can see that with looking at T-cell responses here, but it's ultimately the same thing. All right, so do these fungi matter? Here we're going to supplement with Candida tropicalis or Saccharomycosis fibligera. We're just going to give these mice more of it, these live organisms, by oral gavage. And it turns out that if we just look here in the Dectin-1 knockout mice, when you treat them with DSS, they get a severe disease here, okay, that's exacerbated if you give them Saccharomycosis fibligera, or if you give them Candida tropicalis, okay? So true enough, if you give them more of this opportunistic pathogen, their disease gets worse. Interestingly, if you give them Saccharomycosis fibligera, they're protected, okay? In fact, even the wild-type animals that get a little bit of disease with DSS are protected when you give them fibligera. You can see this in weight loss here, too. Wild-type animals experience a little bit of weight loss. Ramnick had indicated there are like 160 genes that are associated with IBD in people. And so we were very excited to go look. Dectin-1, one of those IBD-associated genes? And the answer is no. All right? But we thought about this a little bit. And, really, we have a Dectin-1 deficient animal and it has no disease. It doesn't get spontaneous colitis. Okay? If we give it colitis, the disease is worse. And so we wondered if really what we could be looking at here is something that modulates the severity of disease. Okay? Unfortunately, at Cedars-Sinai, we had a cohort of people here with ulcerative colitis that had been collected, who had either mild disease that had been managed for many, many years with standard therapy, or very severe disease. And these were patients who had actually progressed to surgery and actually had to have parts of their colon removed. Okay, so a very severe kind of disease. When we looked at polymorphisms in the gene for Dectin-1 in this cohort of people, we found a risk polymorphism that if you carry it, makes you twice as likely to be part of the cohort with this very severe disease, suggesting that there's an association here. Another way to look at this is to look at people with very, very severe disease and stratify them by people with a protective allele or the risk allele, and just watch how long it takes them to progress to surgery from diagnosis. And the people who have the risk allele progress to surgery much faster, okay, suggesting that there may be some predictive utility here. And I'll stop there and just acknowledge the people who've done the work. Ilya is the postdoc who's been working on this project with the help of technicians in the lab. Dermot McGovern, Cedars-Sinai, and his crew were very important for the human genetics. Vince Funari and the genomics core there has become, over the last few years, our master of fungal sequencing, and the Dectin knockout mice come from Gordon Brown. And I'll stop there. Thank you very much.